So, it's very active. We try to engage our membership as much as possible. If you choose to contribute data to this project, you get to choose if you want your data included in any one particular analysis. Maybe you do, maybe you don't, no questions asked.
And a related problem to that is that even if you do write up your negative results that went entirely the opposite of your hypothesis, journals don't want to publish it. And so there's a very serious bias against negative result studies, I think in general, but especially in fMRI.
As with the general fMRI information, this set of questions is not only highly relevant for Cue Reactivity research in all task-based fMRI experiments, task design has an essential influence.
Challenges we have, as raised by Owen. The idea is to combine existing data for secondary meta or mega analysis.
Be a first step, and then people should be at liberty to do the exploratory hypothesis testing. I agree that a lot of people do this massaging. So, but of course, I've never done it.
60 colleagues from different parts of the world contributed to this paper, and it was a really exciting process in terms of running these steps together. I've learned a lot from all these colleagues that you can see there.
Reporting if, when, and how craving is assessed. So the presence, the time point, and the instruments used for craving assessment is essential.
This is something that hasn't really been done yet, but it's one of the future potential directions for fMRI in clinical trials, is to use them for participant selection or stratification.
It is, in my opinion, not unlikely that also in other experiments with low or Cue unspecific behavioral involvement, additional cognitive and affective processes could appear. This is a potential harm for the specificity of findings and could perhaps...
We can see that the most common substance was nicotine, followed by alcohol, cocaine, opioids, and strikingly you can see that the breakdown was not by no means homogeneous across various countries.
So the first one is abstinence and recent drug use.
It is also of note that some stimuli were only used for a specific substance. For example, taste was only used for studies on alcohol.
So what we are going to do, basically, first of all, we will approach those who have already published databases.
So hello, everyone. It's great to have you here. I'm really happy that we are going to have the third webinar in the webinar series by International Society for Addiction Medicine, Neuroscience Interest Group. And I'm really happy that we are going to have lots of really good discussions about biomarkers for addiction treatment development and discussing about fMRI drug cue reactivity. I will have three co-chairs in this meeting. So we'll have Hugh Garavan, we will have Anna Zilverstand, and we will have Antonio Verdejo-García, both of them distinguished figures in the field. And I just want to ask them to just say hello and kind of if they want to make an introduction, and then we will start the program without any further ado.
I'll start. Okay. I'm Anna Zilverstand. I know lots of you. Nice to see you all again. I'm now at the University of Minnesota doing neuroimaging in opioid use disorder and alcohol use disorder. Thanks, Helmut, for organizing this.
Hugh.
Hi, everybody. Hugh Garavan at the University of Vermont. I guess I'm here. Hamid is very good to have made me co-chair. It suggests that I've done work or I've done anything. None of that is actually true. So thank you, Hamid, for organizing this and inviting me. And I guess I will talk a little bit about just the ENIGMA-Addiction group and how we can maybe advance this agenda within that ENIGMA group. So thank you.
Antonio.
Hello, everyone. I'm Antonio Verdejo-García from Monash University in Melbourne, originally from Spain. Same as Hugh, I didn't do anything, so I don't want to take credit for anything in this seminar. Hamed has been incredible in organizing everything. But I will tell you about a little bit more about ISAM-NIG, the Neuroscience Interest Group. We formed this group 4 years ago, and the intention was to have initiatives like this. We wanted to increase the harmonization in the protocols for cognitive and neuroimaging assessment, as well as for cognitive and neurostimulation interventions. And we also wanted to increase the clinical validity of the tools that we generate from neuroscience and to offer more tools to clinicians that are interested in applying this type of ideas. So I think this is going to be a fantastic example of that. Thank you very much, everyone, for contributing, the ones that are really contributing, and looking forward to seeing the presentations today.
Thank you. Now, we also have Alex Baldacchino, the ISAM president-elect, and he's going to be the president in 2 weeks. I don't know, it's just under 2 or 3 weeks. So, Alex, do you want to say hello and kind of...
Typically of Hamed putting me on the spot. So, on behalf of the International Society of Addiction Medicine, which is an international organization that has around 10,000 members, all interested in the field of addiction medicine, with the intention of collecting, evidence, old but hopefully very, very recent evidence in order to change policy and practice. So I welcome this initiative. I remember the time when at 12 midnight, we had this first meeting between Hamed and Antonio around, what the direction of travel would be for the neuroscience interest group. So I'm glad to say that, I think since then we have grown from strength to strength around this subject and other subjects, but particularly around the neuroscience. Thank you, Hamed.
Thank you very much, Alex, for joining us. And many thanks to Antonio. I'm not sure what time is it in Australia right now.
Don't even ask, Hamed.
Yeah, I can imagine.
It's 6 in the morning. It's not that bad this time.
Okay, so I have a kind of lower level of guilt feeling of so that is good. It's because we had meetings with Antonio from like 4 a.m., 3 a.m. in the morning in Australia time. So thank you very much, Antonio, for supporting us all the time. So we are, that is the webinar agenda that we are going to have. So it's going to be the kind of short talks from different colleagues and collaborators in this, let's say, kind of consortium that we are putting together. The first speaker would be Dr. Tanya Ramey from NIDA. And I would like to ask all the speakers to just introduce themselves. So we are not going to kind of officially introduce others. So please introduce yourself. And Tanya, the floor is yours.
Thank you. Thank you so much. And also wanted to thank you for inviting me to speak in this wonderful group, And I'm, very excited to see how enthusiastic you are and what a great leadership you have, and you're all together. And I hope that you will make great strides in what is in front of you and in front of all of us in terms of biomarkers development. I am a medical officer at NIDA in the Division of Therapeutics and Medical Consequences. It's called DTMC abbreviation. And before I came to NIDA, I worked for a long time at Pfizer, where I led early clinical development in neuroscience, specifically for psychiatric indications. And I have done a lot of studies with the biomarkers in translational field in neuroscience. So this is my sort of a background. And since 2014, I am at NIDA and I'm a medical officer in DTMC division. So here on this slide, you see how we want to sort of tackle the biomarkers problem, biomarkers as an issue that is kind of a ripe for development. And I will start from the NOCI, that we have issued a NOCI, Notice of Special Interest, currently, and it's open, still open. You could see the dates and the expiration date on it. And this is sort of an early clinical development piece of the whole biomarker field. And we are looking to develop biomarkers and biotypes in drug addiction. In general, you could take any substance use disorder and you could tackle with it. But more specifically, you'll see what is it about in the next slide. We are looking for discovery, development, and validation of biomarkers. And we're looking also for biosignatures, like sort of in an intermediate phenotype way, to find biosignatures that would be, that where symptoms would be underpinned by neurocircuitry, and we would like to know what kind of biosignatures we could find. And we would like to have projects that could allow us to have subgroups, sort of a biotypes of addiction, and you could find it that those could be not based on the substance being used. As you know, DSM-5, it kind of bases the diagnosis of addiction on the substance being used. But again, this is maybe sort of a more domain-centered, the biosignatures of or subgroups that will be possible to see. And this is a substantial size project, could be, but that's the goal. And as I said, the reconfiguration of substance use disorder classification would be helpful. It would be helpful because we are dealing with a very heterogeneous population in clinical trials. When we look at the populations based on DSM-5, and especially in early clinical development, it's very difficult to elucidate a signal in such a heterogeneous population. I've seen information that for like one substance use disorder, you could have lots of combinations, like thousands of combinations, really. So with that amount of heterogeneity, you could understand that it's difficult to have successful translational studies. So again, just to be more specific, we would be looking at the also peripheral markers, neuroimaging patterns, what we call like neurosignatures connected to symptomatology, neurophysiology data, and wherever you can, you could connect it with the genetics, epigenetics, and genetic risk profiles. And deep phenotyping in terms of like domain structured information. Also for this grant would be very advantageous if you could use big data analytical approaches, machine learning, but it's more for sort of a biotypes, looking into biotypes, machine learning, and such things that could aggregate complex data, big amounts of complex data. And your consortium could do that because you come from all over the world, and so you could amass such databases that could allow for that. And then also projects focused on identification and validation of biomarkers, biosignatures, composites, various composites of peripheral and central markers, distinct molecular profiles, and again, all of that with an aim to identify patterns correlated with the substance use disorder phenotypes, okay? And hopefully learning using the machine learning kind of approaches. Because, there is a lot of interest in that, and it's envisioned that it could provide a breakthrough currently. Again, this kind of a continues in the same way, talking about drug use trajectories, treatment response, also identification of clinical assessment instruments. So I think it's a very inclusive nosing. Now we will be talking, if you could see the biomarkers as a continuum, now we will be talking about other opportunity that is out there for you. And this was recently issued, and this is the RFI, Request for Information, Advancing Biomarkers Research and Implementation for Personalization of Health Services Delivery for Tobacco Use Disorder. This was focused on because it seems that tobacco use disorder is kind of the most advanced in that regard in the field in terms of from that other end, how do you implement and personalize health services delivery based on biomarkers? This one is also open currently, and the application, the response date, you could see in this slide, is December 15, 2020. So you all are invited to consider it. this RFI has that introduction that talks about like how this RFI would think about biomarkers. They would think about it as a behavioral, cognitive, affective phenotypes, neuroimaging signature. That reminds you, I'm reminding you about the NOCI, which is out there, talks about those neuroimaging signatures, biosignatures with other components in it. Genetics and biospecimens. This RFI looks at diagnostic biomarkers, prognostic, predictive, and safety biomarkers. So all the projects that could kind of focus on those, they are eligible to apply. Again, we are requesting this information from clinical scientists. We want to generate interest, and we would be happy to consider those applications. You know, the more active you will be, the more interested will be on our part as well. So we wanted to have the data that are very broad, and we want to kind of, in terms of diagnostic, prognostic, predictive, safety, and how to utilize it in the initial healthcare setting. So in that way, it's a little bit more towards the end, the other end of spectrum of biomarkers per se. And so also, you know, it could be, the applications could be from human laboratory scientists and, you know, from clinical scientists. And it's very broad. And so with that, that was my last slide. And I would be happy to answer any questions. So you could see that we actually took one part, the beginning, the left end of the spectrum, the beginning, the biotypes, the neuroimaging signatures, and then the other end of it as well, as it relates to tobacco use disorder. How would you do those biomarkers to personalize care, for example, in tobacco use disorder? Both are now open for applications, both opportunities. Thanks again.
Thank you very much, Tanya. Thank you very much for such a nice presentation. And what I've realized working with Tanya during the last couple of months and years, she's so responsive and so supportive. I mean, whenever you send an email to Tanya, she would respond really rapidly. And I'm really I really appreciate your support and your kind of help.
Thank you, Hamid. And we are very much looking to receive applications. We want to see activity in the field.
That is great. That is great. Those who have any sort of question, I highly recommend people to post their question in the chat space, so then we can have kind of a better sort of communication and a better flow of the session and being able to ask questions. Then the speakers would also be able to see the questions that you are posting. And we devoted something like 30 minutes at the end of the meeting to be able to give people time to be able to ask questions from the speaker. So that might be the best way to make sure that the timing would be perfectly well managed. If others co-chairs are kind of, they think that we can move forward, we can move for the next speaker. Any specific note, any specific comment? Okay, so let's move for the next speaker, who is going to be Dr. Owen Carmicheal.
Okay, so let's see. I guess I'd start by saying if none of you have ever heard of me or the Pennington Biomedical Research Center, that's fine. We don't really do addiction research. We are mostly an obesity research center, which is where I intersect with this group, I think. I do functional MRI studies of cue reactivity, but they tend to be food cue reactivity studies in the context of obesity and metabolic diseases such as diabetes. However, in 2015, I organized a workshop that brought in a group of people from a fairly wide swath of the pharmaceutical industry to talk about the current state of the art in functional MRI as a tool that can be used in clinical trials and in drug development. And you can see on the lower right here that we published a summary paper from that workshop, which has everything in this talk in it, and I would commend that to you if you want to see more details about how I'm thinking about this question about functional MRI as a biomarker in clinical trials. So here are my disclosures very quickly. I'm doing 2 functional MRI trials that are industry funded. So neither of those are directly related to the contents of the talk. And what I'll do is just to make sure that we're all on the same page, I'll start by giving a very brief overview of functional MRI and Cue reactivity paradigms in functional MRI, just to make sure that everyone is familiar with what we mean when we say functional MRI and food cue and drug cue reactivity paradigms. And by then I'll hopefully spend the bulk of the number of minutes I have talking about how functional MRI can play a role as a marker in clinical trials, and then talk about some of the challenges that are preventing its more widespread application to clinical trials. Okay, so what is fMRI? And this is going to be a 30,000-foot view. So all of functional MRI hinges on the fact that oxygenated hemoglobin and deoxygenated hemoglobin provide very slightly different signals when they are measured in an MRI machine. And what we observe when we do a functional MRI study is really differences in the local concentration of oxygenated and deoxygenated hemoglobin in blood vessels in the brain. And so on the top of this figure is what is depicting neurons that are in a part of the brain that are inactive. And you can see that the little purple guys are deoxygenated hemoglobin and red is oxygenated. And you can see that there's a certain mix of the two taking place there. So when there is a sensory stimulus or a cognitive process or a motor process that started in the brain, neurons fire. We all know about that. But the neurons firing initiates a cascade of cell signaling events that leads to an increase in the relative concentration of oxygenated as opposed to deoxygenated, which is depicted in the bottom part of this slide. And as I said earlier, the MRI signal that you get out from functional MRI differs based on the relative concentration of purple guys to red guys, or oxygenated to deoxygenated. And that's what's called bold fMRI, or blood oxygenation level dependent fMRI. So that's the whole thing. And I think it's very important in the context of clinical trials to remember that what we are really measuring directly is not neurons, it's blood vessels. But what is happening inside the blood vessels is an indirect indication of what is happening to neurons. And so this slide is just to tell you that the set of physiological events that gives rise to a change in functional MRI signals is complex and has multiple steps. So at the beginning, we smell something, we see something, we taste something, we start moving a part of our body, or there's some cognitive process that takes place. That leads to firing of neurons, which causes a metabolic demand for a fuel substrate to fuel that firing activity. And those substrates include glucose and oxygen. Blood flow is how we get those substrates to the neurons to fuel that activity. And that in turn, that blood flow, which brings oxygen to the neurons, causes a displacement of deoxygenated hemoglobin, and then, as I've said, a change in the local MR signal. And just to give one concrete example of functional MRI, imagine that you are put in the MRI scanner, and we go from having the lights on to the lights being off, to the lights being on, to the lights being off. So what we're showing here is an axial view of the brain, and at the top here in A, B, and C is the occipital lobe, the back part of your head, which is also the visual cortex, the part of the brain that primarily processes visual information. And what's being depicted is the magnitude of the functional MRI signal when the lights are on in A, off in C, on in B, etc. And it's being shown in the graph here. You can see that there's qualitative differences in the fMRI signal between time points when the lights are off, and then on, and then off, and then on. And this was one of the earliest fMRI papers that was published in '92. Nothing fundamental about functional MRI in terms of the physiology of it or what we're measuring has changed since that time point in 1992. So that's a brief overview of what fMRI is. So what are cue reactivity paradigms then? Well, what we do in cue reactivity paradigms is that we set up differing epochs of time while the person is lying in the MRI machine. So, and I'm referring to these different blocks of time that are labeled Q, rest as epochs of time. So in one epoch, I call it the Q time period, where the person is either shown visual cues or is provided some other sensory cue. Now, at the Pennington Center, as I said, we do obesity studies. So most of the time what we're doing is showing people junk food as their cues, things like cake and fried food and steak. However, obviously in this setting, you can show other types of food. You can show visual drug cues. You can show visual cues associated with alcohol, pornography, you name it. You can show those visually, but the cues can also be odors, tastes, or tactile stimuli. And again, at the Pennington Center, we've actually done studies where we drip small amounts of delicious milkshake into the mouth and use that as a cue to trigger responses in the brain. So these other epochs are rest, where the person is sitting there nominally not being stimulated by anything. They're just kind of staring at a screen that is in front of them. So if we do this and we present cues and we give rest and we present cues and we give rest, the thing we can do with that is shown in the next slide, where we can compare the fMRI signals that are collected while the person is processing the cues, shown in red, and compare those signals to time points when the person is not processing the cues or when they are resting. That's shown in blue. And shown on the bottom right here is that the great thing about fMRI is that we can measure this at every location in the brain in parallel. You'll hear the term subtraction paradigm for fMRI Cue reactivity paradigms. And that refers to the idea that what we're essentially doing is subtracting on some level the fMRI signal between the red boxes and the blue boxes, or between when the cue is presented and when the person is resting. And in that way, we can draw inferences about what parts of the brain are activated or involved in the processing of these cues and the degree to which they are activated in this way. Okay, so how can fMRI play a role in clinical trials? I think the most important conclusion that we drew from our workshop is that different stages of clinical trials call for different uses of functional MRI and different uses of cue reactivity paradigms. In early phase trials, you've got a lot of questions having to do with your compound of interest. In some cases, you're not even sure that it crosses the blood-brain barrier. And in other cases, the drug industry being what it is, you're not even sure how the thing is doing the thing that you think it's doing. So in this kind of a setting, fMRI studies can support the plausibility of the test article. Now, I'm very careful to tell people that fMRI is not a direct measurement of target engagement. So for example, there's a different technology, positron emission tomography or PET studies, where you can do what's called receptor occupancy PET studies. And so for example, if you have a PET ligand that binds to a dopamine receptor, and you have a test article that is supposed to bind to that same dopamine receptor, you can use the PET technology to tell you what percentage of the possible dopamine receptors are being bound by your test article. And in that way, directly get at how well your test agent is engaging with the designed target. You don't get that with fMRI. Again, you're not directly measuring receptors, you're not directly measuring cell signaling events, you're directly measuring blood flow events that are caused by neural events. However, if you're not sure that the test agent is crossing the blood-brain barrier at all, you can use fMRI to tell you whether providing the drug provides a difference in these blood flow events in parts of the brain that are expected given the hypothesized mechanism of action of the test article. So, and you can see if it's plausible, if the right parts of the brain become activated by the test agent. And you can also look at dose-response relationships. So in early phase studies, if you have an ascending dose type of design, you can see if ascending the dose of the test agent leads to ascending fMRI responses. And one thing that you can do that's kind of nice is see if there's one of the doses of your test agent that leads to a maximization of the fMRI response to it, And that might be one of the many sources of information that you use to decide to settle on one or a few doses to advance onto future stage trials. So in later phase studies, the application of fMRI is actually rather different. So most of the time in these trials, there is an established disease-related fMRI signature, or there is a classical fMRI sign that is shown on the people that are suffering from whatever the disease is. And so I do a lot of work in Alzheimer's disease. There's a particular pattern of fMRI hyper and hypo activations that one classically sees in someone with Alzheimer's disease. And it's especially worse in someone who has more severe symptoms of the disease. Most of the time, what you're doing when you apply functional MRI in this setting is seeing if your test agent brings that disease-related signal back down towards normal levels. And usually, and it's because these studies are quite large and they're usually done at multiple sites. You can also further explore what you think is the hypothesized mechanism of action of your agent. So if you have a very large study that's doing fMRI, you might find that a subset of people respond well in terms of seeing fMRI changes that are associated with your test agent, and other ones that don't. And if you have a large enough sample, you can start to understand why that is the case. This is something that hasn't really been done yet, but it's one of the future potential directions for fMRI in clinical trials, is to use them for participant selection or stratification. And so to my knowledge, I haven't seen any clinical trials to date that have used fMRI as a screen in. So in other words, you do an fMRI study, you calculate some numbers and use that to decide whether the person gets to go into the study or not. Or similarly, using fMRI measures to stratify people into a high dose arm and a low dose arm, or whatever the case may be. But that is one of the directions that, or one of the possible uses for functional MRI in the future as the technology becomes more well developed. Okay, so there are a set of serious challenges that are preventing the adoption of functional MRI in clinical trials in a more widespread way. I'll go through a few of these, although a lot of the rest of this session is going to talk about that. So one, I've already referred to the idea that that there is a complexity in terms of how the bold signal arises in the first place, and there's a complexity in terms of relationships between drug actions and bold signal changes. So it's actually difficult to tie the two together. You know that you supplied the drug, you saw an fMRI change, but there's a whole lot of events that happen in the interim. Also, there's a complexity of relationships between fMRI signals and relevant clinical outcomes. So let's say, for example, that you have a participant who's in the scanner, and when they take a certain drug, their fMRI reactivity to drug cues goes down by 50%. A natural question is how that 50% reduction relates to differences in craving scores or differences in probability of relapse or clinically relevant measures like that. And those relationships are just not that well established yet. There's problems in terms of standardizing the application of fMRI across labs, across populations, across scanner hardware, across software, et cetera. And I think we're going to get into those in the talks in the rest of this session. There are a couple of more kind of community level obstacles in fMRI. One of them is file drawer or confirmation bias effects. This is when I have a very strong hypothesis about how my fMRI study should turn out. It doesn't turn out that way, and instead of publishing it, I put it in the file drawer for no one else to see, or in other words, I bury it. Or the other thing I do is redo the experiment until I get the result I want. And a related problem to that is that even if you do write up your negative results that went entirely the opposite of your hypothesis, journals don't want to publish it. And so there's a very serious bias against negative result studies, I think in general, but especially in fMRI. And also there's false positives because fMRI has such a large amount of data, such a large design space, and such a huge number of different post-processing methodologies that can be applied. that there's a lot of room for statistical massaging of the data, which means that you analyze your data with one set of methods. It doesn't tell you what you want. And so you keep trying different methods, twist, turn, pull, and contort your data until it tells you what you want it to tell you. And this has led to the phenomenon of harking. And I'm not sure if people are already familiar with this acronym, but it's hypothesizing after the results are known. So this is when you analyze your fMRI data, you get some result that you weren't expecting, and then after the fact, you wrap a hypothesis around it. So that, I believe, is the current state of affairs in terms of functional MRI in clinical trials with a focus on Cue reactivity paradigms.
Thank you very much, Owen. It was a really, really nice presentation. The good thing is that we are right on time, so just without any further ado, let's go for the next talk I'm going to present. So my name is Ekhtiari from Laureate Institute for Brain Research in Tulsa, Oklahoma. And I'm going to follow what we have discussed in terms of having fMRI drug cue reactivity as a qualified marker, as an FDA qualified marker in the field of addiction medicine, and how we can get to that point and the challenges that we are facing. As you know, there are many publications, over 300 publications by the end of 2019, using different drug cues to be able to explore brain responses to different sort of drug cues. And over 100 of these trials are basically assessing different sort of interventions in a form of clinical trial. And it's quite amazing that with all these kind of over 300 publications, it seems that we are still in the kind of the infancy, and that we have many challenges to be able to use fMRI drug cue reactivity in a level that could be approved by FDA as a qualified biomarker. And that is the basically one of the major questions, that how we might be able to bring collaborations with all the granting resources that NIDA is providing right now to see how we might be able to get to that level of qualification. But if we want to get to that level of qualification, what we need to have, we basically will have four major challenges to be able to get qualified at that level. And there are specific qualification pathways in FDA if you want to get qualified as a biomarker. And if you read the notice of a special interest that Tanya was discussing about, you can see that NIDA is providing specific support for you to be able to apply for that, let's say, biomarker qualification pathway through the granting money that you are receiving. But to be able to apply for that specific pathway, there are at least four major lines of challenges that you will face. First of all, we need to have harmonized, transparent, publicly available tools that could be used. Second, we need to have pre-registered analysis. As Owen has mentioned, there are a lot of challenges in terms of being able to manipulate the different sort of analysis methods that we use, and we need to have pre-registered analysis pathway and very well defined derived measures. We have issues with reliability and replicability, and also issues regarding validity. And I'm going to kind of talk about these items just in a few minutes, giving you an idea about the challenges that we have. First of all, in terms of the tools that we need to have, these tools should be harmonized, transparent, publicly available. The good thing in the field of drug cue reactivity is that different labs started to put together their databases of drug cues and publish them. During last couple of months, we have several publications with kind of different drug cue databases from tobacco, cannabis, and we put together a paper on our database of meth and opioid cues. And we have published our database of over basically 360 cues with all the cues that are available, with all the assessments and measurements. And we even published the codes and cues for fMRI tasks that are derived from these, let's say, database. And that is basically the practice that we are suggesting people to do, to provide a way to have publicly available fMRI tools that could be used by different groups, and then we can move for the next steps. And then we have challenges in terms of being able to do pre-registered analysis. We know that when we talk about fMRI drug cue reactivity, fMRI drug cue reactivity could activate lots of different brain areas and brain networks. And this is one of the papers that is published by Rita. I know that Rita is here right now with us, and Anna. They put together a really nice paper discussing that basically cue exposure could activate many different networks, and there are specific reasons that why we have these networks and areas getting activated. But then the question is, okay, when we have a map of brain activations and we know all these networks are activated, how we are going to define the specific outcome measure that is going to be our target as something like a biomarker. And even if you put those activations on something like a plot for all those 246 brainstem ROIs, you can see that there are activations in different parts of the brain. From the frontal part, as I show you here, yes, we have them in the frontal part, in the temporal. So we have activation in different parts of the brain. How we are going to define which area or which activation plays a better role as something like a biomarker, or in the process of developing biomarkers in the field of fMRI drug cue reactivity. So there are many questions and challenges. One of the first questions is about the reliability, how much these kind of activations are really reliable. And without reliability, you can imagine that it's really hard to do any sort of test, retest, being able to measure whatever we are measuring in clinical trials through fMRI drug cue reactivity. And you know that there are many challenges for task-based fMRI data in terms of the reliability, and there are recent publications showing that the reliability is really low. We have done a small pilot study exploring different sort of kind of reliability measures, and as you can see here, if we put the threshold of something like ICC of 0.5, there are still many areas that have a specific kind of, they can pass the 0.5 threshold of ICC, which means it's a kind of a reasonable threshold. And even if I show them in something like a Manhattan plot, you can see that there are specific areas that would pass the 0.5 threshold, and even there are areas that would pass the 0.7 threshold of ICC. But this is one of the challenges that we need to discuss about in terms of, okay, what are the derived measures that have higher reliability to be able to use as a biomarker in the next steps. And the other question would be about replication, how much we can replicate the results that we have. For example, in a discovery sample, we have a specific pattern of activation, and then trying that in another replication sample to see, can we replicate the same pattern, and then try it in another replication sample to see if the pattern could be replicated. And also the question of, okay, if we repeat that again in the same sample in the retest, can we replicate the same pattern of activations or not? So there are still challenges for that, and see how we can compare different, let's say, discovery samples and replication samples, and see if there is any level of replication within different, let's say, clinical setting or different clinical population for fMRI drug cue reactivity. Without replication, it might be really hard to be able to sell the idea to FDA as a biomarker. And then in the same context, when we talk about per-registered analysis pathway and defining the derived measures that are going to be our major targets for fMRI drug cue reactivity, we know that there are different networks and different areas. And based on these areas and networks, how can we define the winning area that has both high level of reliability and also high level of signal? So combining these together. In a pilot study, we defined something like 14 different specific areas in the brain. And based on the brainstem atlas, we try to divide these major regions of interest and try to find the areas that are basically winning areas, that they have both high level of signal and reliability together. As you can see here, for each of these, let's say, categories of areas, we try to see, okay, within the, let's say, inferior frontal gyrus areas, what would be the area that would be winning in terms of having both high level of reliability and high level of signal. So then we can define, okay, this is the area that we are looking for among all those 240 areas that we have, and try to use these, let's say, pre-registered ROIs for further analysis or further exploration in terms of which one would be winning as something like a biomarker. So these are the pathways that we might be able to move forward together. But in reality, at the end of the day, the major challenge in the field is about validity. And when we talk about validity, validity is not going to happen without any sort of collaboration. So we need to have collaboration. And that is the discussion that we started within the ENIGMA-Addiction group, discussing about, okay, how we might be able to bring fMRI drug Cue reactivity to the ENIGMA-Addiction working group, and think how we might be able to make a collaborative network of people who are interested to work on this specific area to be able to develop validity evidence. And we started the idea with Anna, we discussed the idea with Hugh and Scott, and we moved forward from that with great support from Hugh. And we put together a pre-registered protocol to be able to run a Delphi study. And you can find the pre-registered protocol in OSF pages. And we basically, based on that specific pre-registered protocol, we have also done a systematic review on all fMRI drug Cue reactivity studies to be able to find experts in the field, to be able to get sure that we will invite all those who have significant publication in the field to the consensus, to be able to bring them together in something like a collaborative network for to make a consensus together. And we started with a steering committee of 14 experts in the field from different, let's say, backgrounds from different countries around the world. And we also invited another group of experts, 41 experts with different backgrounds based on the level of publications that they had in the field, based on the systematic review that we have done. And in 3 steps of consensus development, first of all, we started to develop a checklist of different items that should be considered in reporting an fMRI drug Cue reactivity, because we realized the first step would be transparent reporting and making sure that we are reporting all the details to be able to move towards harmonization in the field. So we started with the checklist development. We had a checklist revision phase afterwards, and after revision, we had a rating phase. And now, based on these basically 3 phases that we did together with a group of colleagues, we put together a checklist of different methodological factors that should be reported if you want to have a transparent reporting. And transparent reporting would end up to make it possible for others to replicate as well. So we have 38 main items. And as you can see, these are the ratings. Some of the items are rated more important, so they are being reported as extremely important or highly important or moderately important. And there are items that didn't receive kind of significant consensus in the field. And also we have developed 75 additional recommendations that probably would be helpful in terms of kind of adding more details. And then we rated them, and as you can see, they have different levels of rating. And my colleagues are going to discuss about them in terms of how we will discuss about all these 7 categories of items and recommendation in the next talks. And the checklist is already available as in 2 versions. One version would have just the items, and we recommend people to report the page number that they are reporting these items. And also we provide people with additional recommendations for each of these items in terms of, okay, if this is the item, then these are the additional recommendations for this specific item to improve the transparency and quality of reporting. So the checklist is designed for reporting, but obviously people can use it for improving their study design as well. And this is the paper that we put together, and we have recently submitted that for basically the preprint in medRxiv, and it is on the review process. And the good thing is that it isn't on the review process. You can read the medRxiv version, And if you have a specific comment or revision, we can still do those revisions in the final version of paper that hopefully will get published somewhere. We are still kind of marketing for the paper to see where it can get published. And as you can see here, 60 colleagues from different parts of the world contributed to this paper. And it was a really exciting process in terms of running these steps together. And I've learned a lot from all these colleagues that you can see their names and their photos here in this page. And as you can see here, we still have remain spaces for you guys, that for those who haven't applied for being a member in the consortium. So we are really interested to have you here. Obviously, we already have Hugh and Scott here, but we still have kind of spots for those who are interested to join us in this consortium. And hopefully we will be able to tackle the challenge that I discussed in terms of making fMRI drug cue reactivity as a qualified biomarker. So we are going to have 5 other speakers who are going to discuss about all these details that we put together in the context of, let's say, the paper, and they will discuss about the technical details that we consider in the paper. And I should thank them all because of their significant contribution as lead authors in the paper and the discussion of the paper. The first speaker would be Jim. Jim, are you online right now with us?
Yes, I am. I just don't have video capabilities at the moment, so you'll have to use your imagination.
Good. Please go ahead.
Okay, thank you. So I'm very happy to be speaking and to be included in the project. It's a great project, and I'm going to be talking just about some of the participant characteristics from the checklist. And I split it up into 2 parts because, as you'll see, there's a whole lot of them. The first part I put into demographics or general characteristics, and the circled items below are the items that passed our most stringent threshold for inclusion into the checklist. So that was inclusion-exclusion criteria, basic demographics, age and sex and gender, as well as psychiatric profile, which includes both diagnoses and dimensional measurements of psychopathology. The items that reached our lower threshold for inclusion were additional demographics like education, intelligence, handedness, and so on. So I wanted to, I only have a couple of minutes, so I can't go over all of those things. So I just wanted to pick one as an example and do the same for the second set, which I'll introduce after this. And so this example is sex and gender as a demographic characteristic. There have been a lot of studies at this point that have demonstrated differences in drug cue reactivity in fMRI studies across a variety of substance use disorders like cocaine use disorder, gambling disorders, tobacco use disorder. And these examples here are 2 papers from the same group in tobacco use disorder and smoking that I thought were a good representation of sex and gender research. So on the left is an example of one of those studies that shows there are differences between men and women. And what they showed specifically was that men had higher cue reactivity in reward-related regions like the ventral striatum compared to females. And they also found that reactivity in men was correlated with craving, self-reported craving, but that was not the case in females. And so on the right hand of the screen, which I think is a great that the field is moving in this direction, not just to look at sex and gender as a statistical moderator, what's different between them, but to try and understand better why these differences may exist. And in this study, they were specifically focusing on menstrual cycle phase as a explanatory variable for variability in cue reactivity in females. And so what they found is that indeed there were big fluctuations in cue reactivity in ventromedial PFC and in putamen, which is what's shown on the screen, in females who had higher activation in the follicular phase of the menstrual cycle, but lower activation in the luteal phase of the menstrual cycle. If you could please advance the slide. So in terms of substance use characteristics, the group and raters, focused on a number of characteristics. The circled ones, again, reach the higher threshold for inclusion. And so that would be current lifetime use pattern severity of the main drug of use within that study, abstinence status, as well as addiction treatment status. The others that reached the lower threshold were route of administration, and then measures of current lifetime use pattern severity of other drugs that weren't the focus of the study in question. So I'm going to talk just briefly a little bit more about addiction treatment status, if you could go to the next slide. And so back in the early days of drug Cue reactivity fMRI studies, in 2004, Wilson and colleagues published a review looking at variability in the brain regions where papers were finding activation to drug cues. And specifically, there seemed to be a lot of variability around the DLPFC and the OFC. And so they tried to explain the variability in drug Cue reactivity across studies by dividing them into whether the participants in the study were treatment seeking or not. And the hypothesis is that people who were not treatment seeking would have a higher perceived drug use opportunity once the scan was over relative to people who were in treatment who wouldn't see that opportunity of use after the scan was over. And in the middle panel here is just an example of a meta-analysis that demonstrated activation universally in the striatum and the ventral striatum. But again, was a good example of how there wasn't consensus around these frontal areas like the OFC and the DLPFC. And so then a little bit after that, the Wilson study and review motivated me to put together a study just looking at the same question. It was not very creative, but just in a single sample and making that comparison within a single study. And we found that, very gratifying, we found the same findings. So there was higher activation to drug cues, and in this case, it was cocaine cues in people with cocaine use disorder, in the DLPFC and the OFC in untreated individuals. And this was treatment status at the time of the study. What's not shown here was also in the study, we looked at stages of change with the Socrates measure, and that just shows also, a further advancement of looking at treatment-seeking status to look at readiness for change, and we found various different activations and differences according to that measure as well. So I'll stop there and thank you.
Thank you very much, Jim, for presentation. And I know that Hugh has another meeting, something like 30 minutes from now. So Hugh, if you want to we can just jump into your talk, making sure that you will present that in something like a convenient way, and then we can get back to here, or we can give you the time whenever that works better for you.
So hi everybody. I'm only going to take about 5 minutes because Hamid and a few others and I have been talking about maybe how we can advance this agenda, sort of harmonizing task-based fMRI within the ENIGMA consortium. So I thought I would just give 2 or 3 slides just describing the ENIGMA consortium. Many of you might be familiar with it, many of you contribute to it, and then we've got very, very broad strokes about maybe a path forward. So if you don't mind, the next slide, please. So I'm Hugh Garavan at the University of Vermont. So ENIGMA, again, many of you may know this, but it's a shorthand for Enhancing Neuroimaging Genetics Through Meta-Analysis. The basic idea is just to give your data a second life, and it speaks to some of the sample size issues that were raised earlier, some of the methodological challenges we have as raised by Owen. The idea is to combine existing data for secondary meta or mega analysis. And so in addition to there just been kind of basic research looking at brain gene correlates, some of the studies that have been published have had over 50,000 participants, which is fantastic for MR. Then there are distinct working groups. And so I co-lead the ENIGMA-Addiction group, again, International Consortium for Multi-Site Studies. We comprise, it's not actually 54, I learned it, 63 research sites across at least 12 countries. And we have, combined pooled data of up to 20,000 individuals. And, again, the large sample size really is, I think, a terrific boon to be able to address some of these reproducibility anxieties in the field. And of course, to have the statistical power to detect brain gene associations. We continue to grow. Please, 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 if you are interested, if you have any data, especially MR data, it's not critical to have genetic data. If you're interested in learning more, please reach out to me. We'll happily chat to you about it. The data itself, that's maybe not fully up to date, but it, gives you a sense, just kind of big picture, that we do have pooled data across multiple different drugs of abuse, including obviously, polydrug use. We are working to improve the behavioral characterization of the participants. That's actually, a considerable challenge, so that we can try to get a handle on polydrug use, psychiatric comorbidities, and so on and so forth. We also have, it's not listed here, but also quite a number of longitudinal cohort studies. Where, so for example, ABCD, Imogen, the Encanda study, developmental HCP. We think this is actually quite an interesting way to go. We might, for example, look at the adults and find, biomarkers, one of the constructs that was mentioned earlier. And then we can go and look in these adolescent developmental cohorts. Are those biomarkers observable in kids who are just starting to use alcohol, or who we know longitudinally go on to become drug dependent? So we can have the different data sets talk to each other. We're busily trying to also ingest large data sets like the UK Biobank, so that we can just continue to increase the sample size. Next slide, please. And so we do lots of stuff. Typically, the primary focus thus far has been on the T1 scans, because if you're doing an imaging study, you're always going to get a high resolution structural scan. So we can look at cortical thickness. We have standardized processing pipelines, standardized quality control procedures. With the cortical data, we can expand to look at things like structural covariance networks across the brain, subcortical morphometrics. We can expand, we have been expanding the last while to include diffusion-weighted imaging, to look at white matter integrity, and of course, resting state connectivity. Almost every neuroimaging study that happens these days includes at least a few minutes of resting state connectivity. So the big challenge here, as you might imagine, is gathering together those data, doing the analysis in a standardized, harmonized manner, and then doing the quality control and so on and so forth. And then all of this, when available, we're trying to build up the number of data sets on the number of participants on whom we have genetic data as well, so for brain-gene associations. So it's very active. We try to engage our membership as much as possible. If you choose to contribute data to this project, you get to choose if you want your data included in any one particular analysis. Maybe you do, maybe you don't, no questions asked. You can propose analyses to work with this large combined data set, and we're trying to encourage as much of that as possible. Next slide. So the big challenge, and then this is the last slide and just the question for today, what of task fMRI? As you might imagine, we start where it's easiest. So, standardized analysis of the T1 scans is relatively straightforward. The other modalities, easier. Task fMRI, of course, is, is the hard one for a lot of the reasons that have been mentioned. But I think, I think this is very doable. The challenges, as I see them, kind of very big picture. Conceptually, I think the work that you've just heard about from Hamid and Anna and Antonio and their colleagues is a substantial step in this direction. Just trying to figure out what the parameter space is, what are the variants across these different tasks. Can we take lessons from the meta-analysis that you just heard about? I think that groundwork is done. So the knowing how one might want to move forward and standardizing across different task variants, I think we're in a very, very good shape there. And when I say we, I don't really mean me, because it's not me who's doing the work. It's Hamid and his colleagues. And then technically, we are very fortunate in that others are thinking the same thoughts. So there is a working group within the ENIGMA family that is focused exclusively on how to do harmonized task-based analysis. And they're looking at working memory tasks and reward-related tasks. And we've already started the conversations with them about how one might go about doing this in an appropriate, standardized way. And then just the stuff in the bottom right corner is absolutely too small to see, and that's to ensure that you don't ask me any tricky questions, because it's about complicated machine learning stuff. But the reason why it's there is just to make the case that working with ENIGMA is complicated. You can very often have lots of imbalances. Some sites only have substance users, some only have controls. But there are ways and means that we have developed to figure this out. There are ways that we can get around the peculiarities of the site-specific confounds. So I think the challenge for task-based analysis, where you know, one might be skeptical from the outset that, well, there are so many variations of the tasks that it just won't be possible, and the particular task version might be confounded with a particular substance using group. I think we can do that. I think we have the wherewithal and the means to do that. So, you know, I'm very, very encouraged. I think this is going to be a big, new, fun adventure for us all. And again, if anyone is interested and wants to learn anything more about the ENIGMA-Addiction group, please, please, please reach out to me. I'll happily sign you up. All right. Any questions or comments?
Thank you very much, Hugh, for such a nice presentation. I'm always inspired by the great work that you are doing in ENIGMA. And also, besides all the things that you are doing, you are still supporting kind of junior scientists like me, kind of adding ideas to what you are doing. And I mean, that's the reason that Anna and me started to discuss about this with you. And I'm really happy that we are moving forward with that. And those who are basically thinking about, okay, how we can bring in our task-based fMRI databases with drug Cue reactivity to this consortium, these are specific discussions that we are going to talk about today. But as Hugh is going to leave us, so I was thinking that it might be a good idea to have his talk first. But we will discuss about what will be the next step. So we have a specific plan that we'll discuss about today, how we are going to move forward and bring in fMRI drug Cue reactivity to the ENIGMA consortium. That is the plan that we have, and I hope that we can. This meeting is going to be the initial step, and hopefully the process would be happening in the next couple of, we'll see, months or years. So we'll see how the process is going to move forward. So those who have any question, please post your question in the chat space. Then we'll have kind of Hugh. He would probably be with us for the next couple of minutes, so he would be able to see the questions in the chat space, and we definitely would be happy to kind of get you connected with other projects within the ENIGMA consortium, as we are doing. I mean, we are already, me, Anna, we are all kind of a part of the ENIGMA consortium, and we contribute our databases to this consortium. We highly recommend others to do the same. So let's be back to the kind of discussions that we had, and going back to the checklist and the technical details that we are putting together for that specific checklist. The next speaker would be Dr. Marc Kaufman from McLean.
Thanks so much. Thank you, Hamid, for including me. This is a great program. I'm learning a tremendous amount, and it's clear that there's an incredible amount to know about this field and to learn and to harmonize as well. I'm going to talk about one of the categories, the general fMRI information category. Next slide, please. Thank you. Yeah. So it turns out that the general fMRI information, you know, pulse sequence, acquisition details, pre-processing pipeline, data processing, data reporting elements of the checklist were collectively rated highest of all of the elements. So you can see they were considered extremely important or highly important by 90% or greater of the raters. I think that's because these are the kind of the factors that really shape the data strongly. And then if you click the slide again, we get the additional recommendations. And again, you can see across this category with a little bit more granular breakdown of the category, very high ratings for many of the elements. Notably, the pre-registration element is up there, but maybe not as high as we need it to get to where we need to be to work in the ENIGMA framework. Maybe this is something to consider, that pre-registration may be a first step, and then people should be at liberty to do the exploratory hypothesis testing. I agree that a lot of people do this massaging. So, but of course I've never done it. So, so next slide, please. What to report in this category? Well, obviously the scanner, the field strength, the head coil, number of channels, pulse sequence, pre-processing pipeline elements. I mean, I could read through this list, but basically what you want to report to be totally disclosing, and so people know what you're doing and what the limitations as well as the strengths of your data are everything. In fact, it seems like there was a strong consensus, especially within this category, but also across all the categories that the experts identified to provide as much detail as possible. So next slide, please. I just want to, in the brief time that we have, show, yeah, that one, thanks. Just a couple of examples. It's not possible to go through how each of the factors, even in the prior slide, could affect studies, but here's just a couple of examples about hardware difference affects the output of the data. So on the left is a relatively new study by Colazzoli and colleagues. It's also posted, available on bioRxiv, and it's been there for, I think, about a year. So I guess it has maybe not been accepted for publication yet. But what this interesting study did was look at 8 individuals who they were able to get to do a rewarding task in a 7T scanner and a 3T scanner. And the idea was, if you optimize parameters to detect deep brain structures that are of interest to addiction researchers and Cue reactivity researchers, including the locus coeruleus, and that's so if we're looking at the top row here of the coronal sections, the locus coeruleus LC, the substantia nigra SN, and the VTA, those are the ROIs from which the activation maps were extracted. And in the lower 2 bars, the bar graphs, we can see the 7T versus the 3T results. First in cortical regions and striatum and brainstem nuclei, and then below in locus coeruleus, substantia nigra, and VTA. So there was an overall response in the left columns, and hard versus easy contrast in the right column graphs. So you can see here very large group differences based on the scanner, not quite reaching statistical significance for some of these measures, but remember, these are N's of 8 only, so far fewer than I think we all appreciate you need in these studies. So if this study were to have 16 or 20 people, I think you would see very large differences based on field strength, and that's of course critical because some of these brain regions are hard to resolve at lower field strengths. On the right, instead of talking about field strength, we talk about the number of channels, or a coil bias effect. All of these scans were done in a large cohort of 77 subjects, comparing the effects of a 32-channel versus an 8-channel coil. And the filtering, the full width half maximum filtering, was 6 millimeters. For those of us interested in the salience network, again, this is resting state, so this is not Cue reactivity. You actually see very little difference based on the number of channels. But for anyone who's interested in visual network, so medial visual network, lateral visual network, default mode network, you see very significant effects of coil. So this sort of study might be very hard to do with Cue reactivity. Basically, unless you have a large enough Cue set to be able to do your studies with a 32-channel and an 8-channel coil. But to me, this sort of study, is something that our field could definitely use, because we would have a better idea than relying on resting state scan data. In any case, there are certainly effects based on the number of channels that are worth considering. And next slide, please. So this is just an example of the spatial smoothing effect of bias on the full width half maximum, and whether or not you use a 6 millimeter smoothing factor or greater than 7 millimeter factor, you get the results in panels A versus B, and then the difference in panel C. So you can see that the location as well as the kind of the volume of activation differs substantially. So these are just illustrative. There are many different elements of this part of the process, and that's why it's so important to report, so people can understand your data, and maybe you haven't replicated what someone else did, but maybe there's a good reason for it. So thank you, and sorry to go a little long.
Thank you very much, Marc. Thank you. Thank you very much for being on time. Torsten, are you online?
Yeah, I joined the ENIGMA-Addiction group about 1 year ago, I think, and it's a pleasure for me to be here for the first time. And I would like to talk about the general task information section of the checklist. Okay, and this slide is animated. Please press the button 3 times again. Okay, thank you. Yeah, as with the general fMRI information, this set of questions is not only highly relevant for Cue reactivity research and all task-based fMRI experiments, task design has an essential influence on analysis, validity, and interpretation of the collected data. By means of the experimental tasks, the research question is transferred in the hypothesis test compatible sequence of stimuli and behavioral requirements. This is done to activate the cognitive and affective brain states of interest and thus to induce that variance in the bold signal on which our statistical analyses are based. Last but not least, to sample the bold response optimally, the task has to consider both the temporal characteristics of the hemodynamic response and the fMRI data acquisition schemes. This is such an important point that the Committee of the Best Practice in Data Analysis and Sharing the Biotypes has already in 2016 fixed the principles for this subject area in its guidelines. The corresponding section in our checklist is widely in line with the Biotypes checklist. It is thus not surprising that the items regarding the general task information is being classified as important as the items of the participant characteristics and the general fMRI information section. 6 out of the 7 items were assessed as extremely or highly important by about 90% of the raters. Within this, the items regarding the general experimental design, the number of task components, and the temporal information received the highest acceptance within the expert panel. In contrast, and a little bit surprising for me, the task code and stimulus sharing were rated significantly less important, although 58% of the channel experts had the opinion that this item should be included in the checklist. This is particularly surprising given the intense contemporary discussion about the replication crisis in life sciences, also in fMRI research. In my opinion, authors should still report whether they have used an open scientific platform to provide task-related data like stimuli or software to the imaging community. This scheme highlights the importance of the items in this report category. I will try to show some important experimental and analytical consequences of design and task-related parameters. In the upper line, you can see the items of this checklist category. And in the lines below, I have listed the cognitive and affective states, the data collection, and the data analysis that are influenced on these items in the checklist, or the items of this category. To find out if the used stimuli are suitable to address the research question, or if the possible contrasts are appropriate for hypothesis testing, the reader needs detailed information about the experimental conditions and the stimuli. In other words, this information is crucial for validity. A further parameter that could influence validity is the requested behavioral engagement, especially in passive tasks. Additional cognitive and affective processes may occur. At the end of my talk, I will provide an example of the effect of unexpected parallel, probably automatic processes on brain response. Furthermore, it is not sufficient to assign the use design to a certain design category like epoch or event-related designs. It is rather necessary to show the exact temporal structure of the task, specifically the order, the onset, the spacing, and the duration of stimuli. Thus, the temporal structure of the task is crucial for the bold amplitude that depends on the duration of the activated mental processes. In contrast, the used task components or experimental conditions are crucial for the location of this effect that depends on the nature of the mental process. The temporal structure of the task essentially influences the statistical analysis, specifically the efficiency and power of bold detection. Due to the central importance of this issue, the next 2 slides are focused on this topic. Please proceed. Next slide, please. Keeping in mind, please put your attention to the left-hand side. keeping in mind that the majority of functional MRI is based on both effects. The nature of this effect, especially its temporal dynamics, has to be considered in fMRI task design. In other words, data collection and data analysis are closely interconnected. Let's assume an event-related design consisting of 7 events. Each single event-related bolt response is sampled by 7 MR scans. In the left upper panel, simulated hemodynamic responses are shown as solid curves for a fixed interstimulus interval and for a variable or jittered interstimulus interval. The dots at these curves indicate the MR sampling points. Below these diagrams, you can see the associated measured time series that are simply superpositions of the underlying and overlapping bolt responses. As can be seen easily, the variance in the jittered design is greater than the variance in the fixed design. Moreover, the set of unique equations for bolt deconvolution is greater in the jittered case. Consequently, bolt detection power and efficiency is influenced by this fact. In the right-hand graphics, power and efficiency are plotted for different stimulus durations and average stimulus intervals. Interstimulus intervals for jittered designs indicated by solid curves and fixed designs indicated by dashed curves. As can be seen, designs with a variable interstimulus interval outperform those with a fixed spacing. At the same time, a main problem of brain imaging becomes visible. For a high bolt detection power, longer interstimulus intervals are beneficial, whereas bolt estimation efficiency is higher in shorter ISIs. In search of a good trade-off between bolt estimation efficiency and detection power, Liu compared different stimulus sequences, and I take this picture from this 2001 publication or paper. As shown in this diagram, fixed designs suffer from weak estimation efficiency and fully random designs have low detection power. An acceptable compromise is given only by semi-random designs, highlighted by the ochre rectangles. However, as already mentioned, the temporal characteristics of a design is not a categorical entity. Reporting should thus mention whether and how the order and timing of stimulus presentation were optimized. Finally, I would like to illustrate the possible effect of task involvement on the result of Cue reactivity experiments by means of a study in pathological gamblers carried out by one of my PhD students. In this experiment, the participants were instructed to respond as fast as possible to a dot presented at the position of one of 2 pictures of the position of one of 2 pictures disappearing immediately before. One of these pictures was an addiction cue and the other a control picture. It should be noted that due to this instruction, no explicit cue processing was required. In this so-called dot probe experiment, the amount of attentional bias is quantified by means of reaction time differences. This attentional bias should then be used as a self-report independent proxy for the severity of addiction and can be correlated with neural cue reactivity in the brain. To avoid anticipation effects, we used 2 different presentation durations for the pictures, 200 and 2000 milliseconds. As can be seen in this activation maps, surprisingly, the well-known cue reactivity brain response pattern was only detectable in the short presentation trials. In long presentation trials, a strong ventrolateral, in contrast, in long presentation trials, here highlighted by a blue to green color-coded map, a strong ventrolateral prefrontal response suggested the influence of additional, very likely unconscious inhibitory processes. We tested this hypothesis by means of a psychophysiological interaction analysis using the right ventrolateral prefrontal cortex, specifically the right inferior frontal gyrus as seat region, and indeed, the resulting pattern of increased functional connectivity during long presentation trials contain many of the brain regions known from cue reactivity research, specifically the ventral striatum and the orbitofrontal cortex. It is, in my opinion, not unlikely that also in other experiments with low or cue unspecific behavioral involvement, additional cognitive and affective processes could appear. This is of potential harm for the specificity of findings and could perhaps result in misinterpretations. Taken together, even without having a specific meaning in cue reactivity research, the task reporting items belong to the most important items in the checklist. To report the experimental task in a complete and standardized manner, authors and reviewers should therefore refer to these items in the checklist developed within this project in the future. Thank you very much for your attention, and are there any questions?
Thank you, Torsten. I think we'll take all the questions together at the end. So I'll continue with I'm Anna Zilverstand from the University of Minnesota, Department of Psychiatry, and I'll talk about what came out of the consensus study with regard to task-related assessments, particular with regards to the craving assessment, both inside the scanner and outside the scanner. So what the consensus study, what the experts agreed upon with a very large consensus of above 80% is that reporting if, when, and how craving is assessed. So the presence, the time point, and the instruments used for craving assessment is essential or highly important. What was lower rated with a rating of about 40% highly important is the technology used to do the craving assessment. So what are the instruments that are available for craving assessments? There's a really nice review from Kleikamp and colleagues that was published last year with regards to craving assessments that have been used to assess craving in individuals with opioid use disorder. And I'm just going to talk about the instruments that they reported that have to do with the assessment of immediate craving. So are you craving right now? And the two, and I will report what is most commonly used in the field. So the two most common instruments used for that currently are the visual analog scale or Likert scales, and you can see two visual examples here of those. And there is decades of research on how to administer these kind of scales and the advantages from the psychology literature. And I'll just very briefly summarize. If you look at all that research, Likert scales are slightly easier and faster to respond to. If scales are explicitly labeled, like the examples I showed in the previous slides, the results are more reliable. A 7-point Likert scale seems to be the ideal in terms of test-retest reliability and validity, but 5-point Likert scales are almost as good. And all of these scales are suited for parametric analysis of the data. So really the outcome of all that literature is that it doesn't really matter that much what kind of scale you use. And there has been direct comparisons, as for example in the graph that you see on the bottom right, where people were administered both types of scales and the outcomes were the same, no matter what the scale used. And what is interesting is that questionnaires can also be used, of course, to assess immediate craving, and there's validation studies. There's a few longer questionnaires. I ordered them in the order of most commonly used. The most often used is the desire for drugs questionnaire, then the heroin craving questionnaire, opioid craving scale, and then the craving heroin inventory. And a lot of these are longer questionnaires. I wanted to just briefly mention them because there is validation studies that have investigated them in terms of the if craving is a multi-dimensional construct or a one-dimensional construct, and what these validation studies have found is that a three-factor solution fits the data best, stating that craving is a three-dimensional construct that has sub-aspects which are desire and intention, or if craving is driven by negative reinforcement, or if craving is driven by the ability to control it. So it is a multifaceted thing that we often assess with a single item scale. And that's important because a lot of these assessments where we're using visual analog scales or Likert scales, we use prompts. And what it turns out that a lot of people use very different prompts, and there's no really any agreement as to with regards what prompt to use. And these prompts tap into different aspects of this three-dimensional craving construct. So for example, the first one, which is most commonly used, is the how much do you feel the urge to use, taps into the desire and intention to use, whereas one of the bottom ones, how much control do you feel, taps into the control aspect of craving. So what prompt you use will actually influence what you're measuring, and you might get different results depending on the prompt you use. So in summary, we want to report the presence of craving assessments, the time points at which we're assessing it, and the instruments, and then what came out of the additional recommendations, and which was rated with high importance above from 80% of experts, links up to what I previously said, because the recommendations were to report not only the time frame in terms of inside versus outside the scanner, but also are you presenting the prompt and the question during viewing the picture or after, and then report the exact wording of the prompt because it obviously matters. Other recommendations were to report the scale used, visual analog versus Likert, the prior validation efforts, and of course also how you're analyzing the rating data. Thank you. So I would like to give it to Amy James, who has looked into pre and post scanning considerations.
Thank you very much, and thank you for including me on this project. Can you advance to the next slide? Actually hit the button 2 more times. Thank you. I also made use of animations. So the first thing that I want to talk about are some of the considerations that received very high rankings in the recommendation for reporting. And then afterwards, I'll talk about some of the ones that were ranked slightly lower. So the first one is abstinence and recent drug use. And this is really important to talk about because several studies, such as the example here by Joe McLean and colleagues, have shown that brain activation to drug-associated cues is actually higher during abstinence relative to satiety. And that lines up with preclinical findings as shown in the bar graph, where you can see an animal's behavioral responding to a drug cue actually escalates over the course of withdrawal. So you get this nice sensitized response. Could you advance the slide? However, the story is not that simple. It's not just that cue responding is going to escalate continuously, but work by Rita Goldstein's group has used EEG to show that while cue reactivity does increase over a period of time, about a month, it then will slowly come down over a year. So it's this idea of abstinence as well as the duration of abstinence that can potentially have a large effect on drug cue reactivity. So another consideration, and this was brought up by Hamed earlier, is his recent work where he measured brain activation in the ventral medial prefrontal cortex. And at the beginning of the presentations, the first one, you see this nice separation of brain activation to drug relative to neutral stimuli. Whereas by the 4th presentation, and this is within a run, so no one's getting in and out of the scanner, what you see is an attenuation, suggesting that there's a habituation of brain activation to drug relative to neutral cues over repeated presentations. Can you advance? But I also want to point out that work by Teresa Franklin at UPenn has looked at brain activation to smoking cues at a baseline scan in that same ventral medial prefrontal cortex region, and then again following 3 weeks of placebo. And they actually didn't see any sort of change in that VMPFC brain activation to smoking cues, suggesting that the duration of time in between cue exposures might impact whether there's habituation or not. I should also point out that the work by Hamed was looking at folks who used opiates or methamphetamine, whereas Terry Franklin's work was looking at nicotine dependence. So it's also plausible that habituation might be drug specific, but it's still plausible that habituation could have an influence on your brain activation. Another thing to consider is drug expectancy. And in this example, all individuals were actually given nicotine, and only some of them were told that they received nicotine, while others were told that they didn't receive nicotine. And what you see is that there's more brain activation to drug-associated cues when there was that expectancy that they had received the drug of abuse. Can you advance? Thank you. And this is true both under abstinent and non-abstinent conditions. So if you look at those top 2 panels that are labeled with expectancy, it doesn't matter if someone is in the abstinent or non-abstinent state, they're still having significantly higher brain activation to drug-associated cues relative to when there is no expectancy of a drug being administered. These are the elements that did not reach that same level of stringent consensus as the others that I talked about in a little bit more depth. The first one still was ranked fairly highly, with about 60% of individuals saying this was highly important. And this is pre-scanning training. And what this means is how much training are you giving a participant before they go into the scanner about what they're doing. And one of the reasons why this was ranked a little bit lower was maybe because this is thought to be a fundamental issue, that before someone goes in the scanner, you usually are going to orient them to what they're going to be doing in the scanner. Cue reactivity also can be a fairly straightforward task that sometimes is passive, so might not need a lot of pre-scanning training. However, depending on what's done, if there's a lot of prior cue exposure to someone going in the scanner, this could affect things like expectation and habituation. So there are conditions under which this could actually have a larger effect on your data. The next one that had the same level of consensus reporting is whether you report doing other tasks in the scanner. And some thought this might not be as important because if you're only reporting cue reactivity, it's thought that there's potentially a singular focus on cue reactivity. However, if other tasks are done, particularly before cue reactivity or potentially affecting some sort of expectancy, it is plausible that these other tasks may impact how the brain is responding to smoking cues. The last consideration that actually received the lowest ranking was post-scanning craving. And what I mean by this is how the staff is actually going to mitigate someone's craving experience that they are having after they've gone into the scanner. And this is different than just measuring craving. And one of the reasons why this was potentially ranked as lower is because it's thought that this is an ethical issue that potentially was being addressed when the IRB protocol was being reviewed, instead of this being a factor that would directly impact brain activation to smoking cues. So while important, it did not have that direct link to affecting the brain imaging findings.
Thank you very much, Amy, for such a nice presentation. And thank you, Anna, for helping me. We need to kind of move forward for the next kind of presentation. We just have one single remain presentation for this kind of panel, and then we will move forward for discussion and how we can do the actual work. So as you can see that we have these. Okay, so this is the slides. Two of my students, medical students, they are going to present what we are doing in terms of running a really comprehensive methodological systematic review on all fMRI drug cue reactivities that are published till the end of 2019. And that is going to be the basis for the reach out process that we are planning to do to contact those who have published databases to see how we might be able to bring those databases into the framework that we are putting together for that ENIGMA-Addiction Cue Reactivity initiative. So Arshia and Mehran, the floor is yours.
Thank you very much, Dr. Ekhtiari, for your introduction. So to give you a broad overview of the structure of the systematic review that we've been doing, here is a table of the search terms of the systematic review. As you can see, there's three groups: fMRI-related search terms, cue reactivity-related search terms, and addiction-related search terms. Ultimately, the final syntax on PubMed on January 2020 yielded 677 studies. As you can see, this is the Prisma flowchart of the study. The 677 obtained studies, together with one kindly suggested study, were reviewed, and ultimately, after title and abstract screenings and the full text screenings, we were left with 331 studies. Ultimately, most studies that were excluded were either not fMRI studies, or did not include the relevant populations, or did not have a cue reactivity related paradigm. On the next slide, you can see the geographic distribution of the studies that we included. As you can see, the expectedly United States comes out on top with 182 studies in our database, followed by Germany, China, the Netherlands, Korea, and so on. Further breaking down on the next slide, these studies by the main substance that they were investigating, we can see that the most common substance was, by the substance or behavior, we can see that the most common substance was nicotine, followed by alcohol, cocaine, opioids, and strikingly, you can see that the breakdown was not by no means homogeneous across various countries. And it appears that, for example, in the United States, the vast majority of studies were on nicotine. In Germany, most were on alcohol. In China, opioids, and so on. And this is the yearly breakdown of the studies, except for an unexpected dip on 2016. We can see that the general trend of publication has been steadily going up. And as you can see, the most commonly investigated substance or behavior in our database was nicotine, followed by alcohol, then cocaine, opioids, cannabis, video games, methamphetamine, gambling, and some studies had multiple substances or behaviors. I'll hand over the presentation to my colleague Mehran so we can go over some more detail and discuss the future directions.
Hi everyone, I'm Mehran Zadeh and very happy to be here. About the stimuli utilized in the fMRI reactivity studies, as expected, most of them use pictorial stimuli, but other stimuli used including videos, audio scripts, and so on. As was mentioned, the most common substance was nicotine, but as you can see in the right figure, it is of note that the stimuli used for this substance are less diverse than, for instance, cocaine. It is also of note that some stimuli were only used for a specific substance. For example, taste was only used for studies on alcohol. For the general study type, it is found that researchers were more interested in block designs paradigms with a dominance of 62%. Here is the shape of our database, and we are now extracting more than 70 items, and we hope to finally get it done. And in the end, I should thank Hosna Tarakkoli and Panyan Rafei for the great contributions in the systematic review process, and Mohsen Ebrahimi for his help in illustrating these figures. And also, as you've seen in the previous slide, the database is readily available and you can have access to it. Thank you so much.
Thank you very much, Mehran and Arshia. I mean, as you can see, these two brilliant young champions, they are really kind of substantial for what we have done in terms of putting the systematic review together. And I'm trying to see how we can have a next generation of addiction scientists in the field. I can see lots of really brilliant minds that I'm really hopeful about them to be shining stars in the field in the future. So as you can imagine, based on this database that we have, we are going to reach out to those who have published the databases and see, as the initial first step, how many people are willing to share their databases and how we can move forward with the next steps and start to plan for the analysis pipeline. Now, I'm going to hand over the process to Anna and Antonio to manage the discussions and things that we are going to do for the next 15 minutes to see how we can have feedbacks from others to see how we can move forward. Anna and Antonio.
I saw that there's a comment from James Bjork that the rapid habituation of the striatal response over time is really striking, and it reminds him to in anxiety disorders, for example, it's known that amygdala similarly habituates very rapidly. And I think this brings up something that is related to study design is there's no convergence or idea or even investigation at this point as to how long these tasks should be. And I think that might be an interesting first goal for a collaborative project to figure out how, for resting state at this point, we know there is a few minutes that you need. Some people say 5 minutes, some people say 10 minutes, some people say 20 minutes, but there is like some investigations into this, right? For drug cue reactivity, there isn't. I don't know if anyone wants to comment on that. I see that Betty Jo is posting a question. Do you just want to speak up?
Okay. Hi. Yeah, I was just wondering, I really appreciate Hamed's publicly available pictures and things, but one of the things that we were told when we first got into this field and asked somebody across the country who was helping us to share some cues with us, and she said, no, it differs wherever you are, and if I give you cues, they're not going to be relevant to the people in your area. You got to develop them for the particular area. And that's true even within people. I mean, it's fairly standard in cigarette studies to, you don't give people, you present their brand to them as the cue. So I'm wondering if we should be thinking more about attention to how to appropriately individualize cues for individual subjects, rather than all the emphasis being on getting a standard set of cues to present to people.
One of the comments about the specificity of the cues, we already have a specific item in terms of the tailoring and how we might be able to do the tailoring. But one interesting point is that the database that we developed with opioid and meth users is quite striking that we received email from people in China, and it seems that they are using the database for Chinese population in different parts of their country, and it seems it is working. So it seems that there is a level of universality, at least for some of the drug cues, that could be used. But obviously, we recommend people to redo some of the validations to get sure that the database is working on their own, let's say, cultural background.
Yeah, and the solution might be to just join different databases, right? So I mean, I've also tried I've used Hamed's database, but I've added cues with African Americans, for example. But if everybody would add their databases together, there would be a greater variety to choose from for everybody. So that might be a...
Yeah, should we be thinking about asking people how they are validating the cues that they're using within their population? Was that... Yeah. Discussed in the checklists?
Yes, I think so. Right, Howard? Yes.
Yes. There are specific recommendations in terms of what are the validation things that people can do and what are the assessments that they can do for the cues. There is a sort of kind of comprehensive part in that piece of the manuscript, and there are specific discussions about that. And I highly recommend you to see that part. And if you have any additional suggestion or comment to that, just feel free to post it for us. As I told you, the manuscript is still in the review process, so we still have time to add any additional suggestion to that. Not to the items, because basically the items and recommendations cannot be changed because we have rated them and they are going to be there, but we can change things in the discussion part. And meanwhile, the plan is updating the checklist and the items and recommendations that we have every something like 5 years. So we are going to be back and reviewing the checklist in 5 years and developing the new version. Right now the version is 2020, so we hopefully will have the version of 2025. So please know if there is any specific comment or suggestion in terms of revising any item or adding any other additional item to that. We definitely will have it, and we have already started to develop a list of the recommendation or suggestion for the next version of the checklist.
Someone in the chat is asking if we are suggesting to report route of administration in the participant characteristics. I don't remember.
There are specific, it is a really comprehensive checklist. We have, as I told you, 38 items and 75 recommendations. So there are minor details inside, but we have discussed about comorbidities and psychiatric comorbidities. And I mean, you need to, and the interesting part is that because we have started with a really simple version, and the version evolved over the process of time with adding different comments and discussions. Even I clearly remember that we had specific comments, but we finally, I mean, in the steering committee, we decided not to include that. So if you have a specific even interest to the specific items, you can go back and see different versions, because we put different versions in the OSF web page. So you can see the initial version, the version that has been developed afterwards, and so you can see the process of evolution for this checklist. And there are items that, as you can see, are basically evolved in the process of revisions and addition to the checklist.
So there's some agreement that the How long the task needs to be is important for obvious pragmatic reasons. I don't think there is such a study out there, right? So
one thing Anna, that might be interesting in terms of the timing, we have already started to discuss about what are the questions that we can answer if we have all the databases that are collected in the field in one single, let's say, repository. And probably one of the questions that we can answer with that kind of collective database is the question about time, because people are collecting kind of tasks with different timing. Even when they use event block, they have event block in different timing. So we might be able to move forward and do some analysis in terms of something like considering a temporal dynamic analysis or sliding window, or considering different blocks and considering the effect of time. So probably one of the questions that we can answer with the collective database within the ENIGMA-ACRI consortium would be the effect of timing. And that might be opening another potential in terms of if you have a specific question that you think that it could be answered if you have the entire database of all the studies that people have published so far, please share the questions with us. So we are already collecting a list of questions. That could be basically the list of the projects that people can do when we have the large database of all these studies that have been done before.
And there's no additional questions in the chat. Is there anybody else who wants to speak up?
It would be nice if all the people who are online right now with us just turn on their videos to see, faces, see how many people are not in pajamas and they are,
And you could probably turn off the screen sharing, Howard.
Yeah, of course I will do that. Yeah. And hello, Jim. Okay, good. We have the good thing. We have a limitation in terms of the number of videos that people can turn on. So I know that we have already reached to the limitation of number of cameras that we can have. But it is great to have all these brilliant faces attached to the names, and so we are hopeful to be able to move forward together.
Somebody is asking if there will be an opportunity to add Cue Reactivity data at a later stage. I would say yes, right? This is an evolving project, just like ENIGMA always has been an evolving project. Bedtime in Pakistan. So, Hamid, what do you think? How are you going to keep everyone updated on the next steps?
So what we are going to do, basically, first of all, we will approach those who have already published databases, but please proactively contact us if you feel that you have a database that you are interested to share. Just send us an email. Right now, we are just in the process of just asking people if you are basically interested or not. We will discuss about the technical details and how that is going to be happen and how we are going to kind of solve some of the challenges in terms of the information transfer agreements. There are some technical challenges that we'll discuss about that. But those who are basically willing to share something to the consortium, just send us an email that I'm basically interested. If everything is going to be 100% fine, I will be willing to share something within the kind of consortium. There might be people who feel that, okay, in the best case scenario, I'm not going to share anything. So that, we are not calling those people. Those people who might be interested to share something in the database, whether it's going to be a Cue database, a task of an fMRI task, or 1-2 subjects that they have collected before. Whatever they are willing to share, please contact us. And I would ask Tanya to share our email, the ENIGMA-ACRI email, and also the ISAM-NIG emails. And also we have a Twitter account with both ISAM Neuroscience Interest Group and also ENIGMA-Addiction Cue Reactivity. So if my colleagues and assistants would just share those Twitter accounts, please follow us on Twitter. So we will post whatever we want to do for the next steps in the Twitter channel. And meanwhile, we are going to post any new publications in the field as well. So we are monitoring new publications in the field. So if you have any specific new publications or even a preprint that you want to be kind of introduced to the broader audience of addiction cue reactivity, we definitely will be happy to share that with the community. So please share whatever you have. And even in terms of the experiences, right now in some of the networks that we have, we just share one single slide that we found interesting in the network. So please share whatever you have, and I mean, we definitely would be happy to move forward with that process. So it seems that we are on and Antonio, it seems that we are almost at the end of the meeting time. Any other conclusion from Anna and Antonio?
Oh, I just wanted to tell Mila to contact me personally because I think his question is related to my paper. And then also say that Parnyan has put the email and also the Twitter account into the chat so that people can follow us. And thank you so much, Hamid, for organizing this.
Thank you very much, Anna and Antonio, and everyone who has contributed to this process. Thank you very much, everyone. Antonio, any final comment from your side?
Yeah, well, what I would say is, first of all, I would celebrate the incredible scientific quality of the webinar, and also how dynamic and transparent this community is. I think as soon as we pose questions and challenges, we've seen people participating and bouncing ideas. So I feel really encouraged by this philosophy and this attitude, and congratulations, Hamid and Anna and Torsten and everyone that has been really actively involved in developing this consensus. What I would like to do is to encourage you. I think like this consensus is going to be incredible in terms of improving harmonization, reliability. I would take the opportunity to encourage you to think about validity, and especially clinical validity, and start thinking on how we can leverage this effort and try to see how, for example, Cue reactivity studies then correlate with things like experience sampling of craving in daily life, and clinical prognosis type of measures. I think that will be a massive step ahead in the field if we can link these imaging measures with clinical measures. And the other thing I would encourage you to do is, obviously this is a massive step ahead in terms of like having a harmonized protocol to measure Cue reactivity, which can be a proxy of incentive salience. But now that we're in the mood, we should go and try to go for the some of the other key constructs that are relevant in addiction. So what about developing some harmonized protocols for executive functions, for decision making? So it was great to hear today that as part of ENIGMA, there is also a group that is working on harmonization of task-based fMRI consensus. And I think we should interface with that group and try to develop these comprehensive protocols to try to better understand phenotype, develop these biotypes that Tanya was highlighting at the beginning. So that's it from me. I would encourage everyone to contact us and to be active in the ISAM-NIG group. This is a lovely community. So really happy to be learning from you guys.
Thank you very much. Have a good day.
Thank you, everyone.
Thank you.